Hello, everyone. This is Dr. Sami from the Biostatistics Learning Group, and here we'll talk about uh, correlation and regression in an easy way. So let's get started. Uh, Correlational and predictive techniques. So biomedical research often seek to establish whether there is a relationship between two or more variables. For example, is there a relationship between salt intake and blood pressure, or between cigarette smoking and life expectancy? These methods used to do this are correlational techniques from the word correlation, which focus on the correlatedness of two variables. There are two basic kinds of correlational techniques. One correlation technique is known as correlation. The second one is known as regression. So correlation is used to establish and quantify the strength and direction of the relationship between the two variables. Whereas regression is used to express the functional relationship between the two variables so that the value of one variable can be predicted from knowledge of the other. So it's about prediction of the other one. So correlation simply expresses the strength and direction of the relationship between two variables in terms of a correlation coefficient signified by R small. Values of R vary from -1 to +1. The strength of the relationship is indicated by the size of the coefficient, while its direction is indicated by the sign. So the strength of the relationship is indicated by the size, whereas the direction is indicated by the sign. So a plus sign means there is a positive correlation between the two variables. High values of one variables, such as salt intake, are associated with high values of the other variables, such as a blood pressure. A minus sign means there is a negative correlation between the two variables. High values of one variable, such as cigarette smoking, are associated with low value of the other, such as life expectancy. If there is a perfect linear relationship between the two variables, so that it's possible to know the exact value of one variable from the knowledge of the other variable, the correlation coefficient R will be exactly ±1. If there's absolutely no relationships between the two variables, so that it is impossible to know anything from one variable on the basis of knowledge of the other variable, then the coefficient will be around 0. So a coefficient beyond plus or minus 0.5 are typically regarded as strong, 0.5. Whereas coefficient between 0 and zero 0.5 are usually regarded as weak. So scattergrams and bivariate distributions. The relationship between two correlated variables forms a bivariate distribution, which is commonly presented graphically in the form of a scattergram. The first variable, salt intake, cigarette consumption, is usually plotted on the horizontal x-axis, and the second variable, blood pressure or life expectancy, is plotted on the vertical y-axis. Each data point represents one observation of a pair of values, such as one patient's salt intake and blood pressure. So the number of plotted points is equal to the sample size n. Figure 4.1 shows our different scattergrams. Determining a correlation coefficient involves mathematically finding the line of best fit to the plotted data points. The relationship between the appearance of the scattergram and the correlation coefficient can therefore be understood by imagining how well a straight line could be or could fit the plotted points. In figure 4.1a, for example, here, it's not possible to draw any straight line that would fit the plotted points at all. And the R here would be 0. It is all over the place, the dots here. Whereas the correlation coefficient here is approximately 0. In figure 4b, a straight line would fit the plotted points perfectly. So the correlation coefficient is 1 plus 1. Figure 4.1c would indicate a negative or strong negative correlation. And here it's minus 0.8. And figure 1d shows a weak positive correlation of about plus 0.3. So we said that weak is from 0 to 0.5, strong is plus 5 toward 1. So types of correlation coefficient. The two most commonly used correlation coefficients are the Pearson produced moment correlation, which is used for interval or ratio scale data. And we know that ratio involves true zero, interval is not. And Spearman rank order correlation, which is used for ordinal scale value, so ordinal of order to it. So Pearson, interval or ratio, Spearman, ordinal. The later is sometimes, which is Spearman, symbolized by the letter P, rho. Pearson R would be used, for example, to express the association between salt intake and blood pressure, both of which are ratio scale data, while Spearman's uh, correlation coefficient would be used to express the association between birth order and a clear or a class position at school, both of which are ordinal scale data. Both these correlational techniques are linear. They evaluate the strength of a straight line relationship between two variables if there is a very strong non-linear relationship between the two variables. The Pearson or Spearman correlation coefficient will be an underestimate of the true strength of the relationships. Figure 4.2 illustrates such a situation. Here it is. This is a strong non-linear relationship. A drug has a strong effect at medium dosage levels, but very weak effects at a very high or very low doses. Because the relationship between dose and effect is so non-linear. The Pearson-R correlation coefficient is low, even though there is actually a very strong relationship between the two variables. Visual inspection of scattergrams is therefore invaluable. Invaluable in identifying relationships of this sort. More advanced nonlinear correlation techniques can be used to quantify correlations of this kind.